So, good evening, guys. Uh, Today we'll be starting with the discussion of week 2 for the course in experimental biology or biotechnology. So, do you have any doubts from the last lecture what we have discussed? So, I shared the presentation on a drive link that would be accessible to you all through the website, and I was not aware that it was not in the accessible mode, so I just changed the settings now, so you can go through and access the presentation. And if you have any doubts, we can discuss now, or we can move forward with the discussion. Okay, if not, then we'll start with the today's discussion. So from the last lecture, we have left one question that was left to be discussed. So the question was, what will be the pH of a solution resulting from a mixture of 200 ml of zero point 0.1 molar NaOH and 100 ml of zero point 0.3 molar acetic acid? Okay, so anybody tried it at the home? So we can discuss the answer. Okay, so as in the last lecture also I discussed that how we'll solve this question. So first what we'll do, we'll write the equation. Okay, that how the reaction would occur. So this is NaOH and this is the acetic acid. When we are reacting with this, so we'll form sodium acetate and H2O. Okay, so we have to calculate first the concentration of this particular thing so that we can calculate the concentration of this acid that is left. Okay, then we can calculate the pH of the resulting solution. So how we'll do this? First, we'll calculate the moles of NaOH. Okay, so that would be zero point 0.1. This is because of the ml to liter conversion. Okay, so this is the conversion factor that we have. So this would be 0.02 moles. Then we have the moles of acetic acid that were used initially. Same concept here applies. Okay, so why we have applied the molarity into volume? Because the formula for molarity is number of moles of solute by volume of solution in liters. Okay, so we have to calculate the number of moles, so we'll multiply molarity with the volume of the solution. So here it will come 0.03 moles. Now what we'll do, we'll check the number of moles of acetic acid that are left. Okay, so how we'll calculate that? Because these were the total moles of acetic acid that were present initially, then these much number of moles would be reacted with NaOH to form the sodium acetate. Okay, so now the leftover moles of acetic acid will be zero point 0.01 moles. Okay, now from this we'll calculate the molarity of acetic acid that is being remaining. Okay, so this would be zero point 0.01, and the total volume of solution now will become 300 ml, because 200 ml was NaOH and 100 ml was acetic acid. Okay, so now the total volume would be 300 ml. So this would be 0.3. So now the total molarity of the solution would be 0.033 molar, and then we'll see the number of or the moles of sodium acetate formed. That would be because we have these many moles, so we are considering that these many moles of sodium acetate would be formed. So this would be 0.02 and zero point 0.3 would be the volume. So this would be 0.067 moles or molar. Then we'll put the equation in the Henderson-Hasselbalch equation. That would say acetate ion and the acetic acid that is remaining. Okay, so now the pK value of the acetic acid solution is around 4.76. Okay, this would be log acetate. The amount of acetate we have is 0.067 and 0.033 molar. So this is the molar ratio. Okay, then after calculation it will be around log 2 point something. Okay, so log 2, as I told you in the last lecture also, log 2 value will be 0.301. Okay, so this would be nearly zero point 0.3 something, because here the 2 is there. So after that something would become that comes to the other digit of after 3. So this would be around 5 point something. Okay, so this would be the final pH of the solution. So that's why the correct answer would be answer 1, that is 5.07. So please tell me if there is anything anyone would have not understood in this question. So please let me know if you need to understand this question again. If not, then we'll move on to the next question that is related to the week 2 of the experimental biology. So one thing that like which was not explained in the last lecture also, and I think it would be better if I'll explain it now. So that is the concept of isoelectric point. So we'll start with that, and then we'll move further to electrophoresis. So the question is, isoelectric point is the pH at which the protein is. The options are has no net negative charge or no net charge, will not move in the electric field, has equal positive and negative charges on it, and all of the above. So can anybody guess the answer of this particular question? Okay. Any more answers? Okay, someone said 3 also. Okay, any more answers? So, I hope everybody here is well aware of what is an amino acid. Okay, so I'm drawing a schematic of an amino acid. Please tell me whether it's right or wrong. Okay, so is this form exist in the living world of of a particular amino acid? Can anybody say yes, no, anything that you know? So is this schematic of an amino acid is right or wrong? Please let me know that first. Okay. Any more answers? At least comment so that I'll be able to know whether you are understanding or not what I'm teaching. So, what happens inside a human body whenever we see an amino acid? Okay, so inside our human body, the amino acid do not flow in this form. Okay, this is not the actual form of an amino acid. Although we call it as an amino acid, but the actual form is this. Okay, it's always charged, whether it's moving inside the body or we are using it in a solution form. This is the original form of amino acid that we see. Okay, this is the functional form of an amino acid that we see. So, what happens? There are two chargeable groups. First is the amino group, and this is the carboxyl group. Okay, this is the alpha carbon which is connected to all the four groups. This is the hydrogen and this is the side chain of an amino acid. So what happens? These charges are basically gives the actual charge or net charge on the protein, on the amino acid, or when it is connected with the peptide bond to the protein. Okay, so what happens sometimes when the pH of the solution varies with the situation or with the solution? So when pH of the solution varies, these charges will have a, like the net charge on an amino acid changes. Okay, either it becomes negative or it becomes positive. So sometimes this is the neutral form of an amino acid where it will have both the charges. So the net charge on the protein would be 0. Okay, because it has both plus 1 and minus 1. So if we'll add both, then that would give 0. So the concept of pi comes when the net charge on the protein is zero, so this is called as the isoelectric point. Okay, so the pH at which the net charge on a amino acid or on a protein is zero is called as the isoelectric point. Okay, that means it will have both positive as well as negative charges in equal ratio. That is basically how the uh, protein will be formed. So yes, it has no net negative charge, no net charge, and the equivalent negative and positive charges on it. And then what happens when we run it on a electrophoresis unit? So as you all would have read by now, that electrophoresis means movement in an electric field. Okay, so when you are starting with a low uh, charge potential to a low potential to a higher potential, so that in that way the there would be at some point the net charge on the protein would be zero. So at that point it will not move into any towards any of the electrodes. Okay, neither to the cathode nor to the anode. So at that point it will be formed as a band. Okay, so that is why at this particular pH, when the protein has no net charge or the net charge on the protein is zero, at that particular point, the protein will not move in an electric field. So it will be stayed at that point itself. So this point is also correct. So that means the answer is all of the above. Okay, so does everybody understood the concept of isoelectric point or the isoelectric pH of a protein? Just 1 minute. So, talking about alanine, okay, it's a hydrophobic amino acid. So it has methyl group as its side chain. Okay, so because there are two charge groups in alanine, it will have two pK values of both the groups. So 2.34 will be the for the acidic group and 9.7 will be the for the basic group. Okay, so what will be the PI? So when we talk about PI, so how is PI calculated? It is the mean of the two pK values. Okay, and in some cases there would be three pK values also for an amino acid. In, for example, in charged amino acid, either it's positively charged or the basic amino acids or the acidic amino acids. Okay, so this is the general formula of an PI. So here there two pK values are there, so we'll just take the mean of both. Okay, so this would be around. Okay, so the correct answer would be option number 4. Okay, so does everybody understood the question and how to solve it? Okay, so moving on to the next question. So what will be the PI of the glutamate? Here the pK values of all the three charged groups are present here. So what will be the PI of glutamic acid? So someone said first. Okay. Any more answers? Okay. So in these kind of questions, there is a trick. Okay, rather than a concept. So what happens whenever there are three pKs given for any amino acid, being it a basic or an acidic amino acid. Okay, so the basic amino acids would be histidine, arginine, and lysine, and acidic amino acids are two only, that is D and E. So aspartic acid and the glutamic acid. Okay, so in all five cases there will be three pKa values. Okay, and in all the three cases, what will happen? There will be two pKa values that are near to each other. Okay, so likewise, in this case, these two are near to each other, and this one is the farmost. So, what we'll do, because in the PI formula we have to put only two pK values. Okay, so here we'll put 2.19 and 4.25. Based on this, so this will be 3.22. Okay, so the pI of this particular glutamic acid or the glutamate is 3.22. So the correct answer is option number 3. So any doubts? So in any of these cases, in any of these five cases, basically, we'll use the two pK values that are there that are near to each other. Okay, we cannot take any other, like for example, if there are three pK values, it will not be added up. Okay, so what we'll do, only the two pK values are required in this particular for the calculation of isoelectric point. Okay, so now why we are selecting only two values that are like near to each other, because the side chain that we have, yeah, that's what I'm explaining right now. So why? Because the side chain that we have, okay, for example, in case of basic amino acid, the side chain pK would be more than 7. Okay, now if we calculate the more number of positive charge or the like the concept of pH in case of a basic amino acid, so it will be more towards the positive charge. Okay, so that would be reflecting the pK values of greater than 7. Okay, so that is why the pK values that are near to each other would be more preferable to reduce the pi of that particular amino acid. Okay, is this explanation fine? uh, or any doubts in this particular concept? Okay, so I talked about the case for basic amino acids, right? Now in case of acidic amino acids, what will happen? The pK value of the side chain, okay, that would be less than 7. Fine. So what will happen? Actually, it's acidic in terms of the release of H plus ions, right? So as we discussed in the last class also, according to Arrhenius or any concept that we study, the acid-base concept, anything that releases H plus is a acid. Okay, and as per the pH scale, the acid have a pH of less than 7. Fine. So the side chain of an acidic amino acid would have a pK of less than 7. Okay. Now, what would be the case if we have to calculate the pI of that particular acidic amino acid like glutamate or aspartate? So, in that particular case, we'll consider only the pK values that are less than 7. Okay, so if we'll consider the case where the the basic pK is also there, that is for the amino group. So in that case, the pH or the pI would shift around 7. So that would be not the actual representation of an amino acid. So am I clear?
Yes, sir.
Okay. So any more doubts in this concept? Because this is an important concept and it would be asked in any of the examinations that you follow. So if you have any doubts, we can discuss right now. So if not, then we'll move on to the next question. So this concept would be helpful to understand this particular question also. So glutamate has a pK value of these three, like I have discussed in the last question also. At what pH it will be positively charged? Please guess and think and answer. I would rather say. Okay, some people said 4. Okay. So, what is the structure of a glutamic acid? Okay, so this is the side chain of a glutamic acid, and this is the charged group. That's why it's an acidic amino acid. The pK of this particular amino group is 9.67. For this, it's 2.19. Sorry, 4.25, and for this, it's 2.19. Okay, so now what will happen if now one concept we learned is how pH varies and how it affects the pKa values. Okay, so consider, like, this is the glutamic acid case. Okay, so if pH is less than the pKa value, it will be positively charged. Okay, if pH is greater than the pKa value, it will be negatively charged. How? So, as you know, if pH is less, that means hydrogen ion concentration is more. Okay, so if there is a COO negative group, then that would attract H plus ions and it would form COOH. Okay, so the negative charge on the carboxylic acid group is gone. Now we have the amino group. Okay, so it's a basic in nature. That means it will attract hydrogen ions. Okay, so when H plus will be more, it will attract and it will form a positive charge because it has gained the H plus ions. Okay, so this is called as an amino group. Now this will provide the positive charge on that particular amino acid or at that particular group that we are talking about. Similar is the case when pH is greater than pK, that means at that point of time H plus ions is low. Okay, so the COO group that we have will donate its H plus ion to the solution because the solution needs H plus ion to maintain the pH. Okay, and similarly when we have the NH3 plus, so in this point also because the H plus ion in the solution is low, so this would also donate. So now this COOH after donating H plus will become COO negative. So it would provide the negative charge on that particular amino acid or that particular protein. Okay. So now considering the case of aspartic acid because it has 3 pKa values. So at pH less than 4.25, it would be positively charged. Sorry, it would have no charge. Okay, at pH less than 4.25, this would have no charge. Okay, the charge would be 0. At pH less than 2.19, it would be 0. Okay, and for this also, if the pH would be less, then it would be positively charged. Okay, am I clear with everybody? So now for this particular amino acid or glutamic acid to be positively charged, both of these groups also would have no negative charge on them. Okay, so for that pH less than 2.19 would be preferable to provide a positive charge on this particular amino acid. Okay, so that would be uh, okay, so no option is there, but this would be nearly this pH. Okay, this would be less than 2.34 rather. Yeah, so this pH would be less than 2.19. That would provide a positive charge on the glutamic acid. Okay, so yeah, the options are not right in this case, but that would be less than 2.19. If this option would be 2, then that would be the correct option. Okay, so anybody has any doubt, or shall I explain it again? Okay, just 1 minute. Yeah, you can consider also that thing also, because now I was explaining the concept that how if less than pH the positive charge will be there. Okay, so yes, that would also be the same concept that will apply here. Okay, so less than, if the pH is less than the pi, that would also impart the positive charge on the particular amino acid. So this is the same concept, but I'm explaining it for different groups so that it would be easy to explain. Okay. So now what we'll do, for example, here we have a simple amino acid. We'll talk about, okay. So now what will happen? Consider this as the its pK is around 4.25 per se. Okay, nearly 4 is the pK for the carboxyl group, and its pK is around 9.67. Okay, this is a generalized thing, and it will vary depending upon the side chain that the amino acid have. Okay, so now what will happen if considering the case pH is less than the pK value? First thing, then what will happen, because at low pH, that means the hydrogen ion concentration is really high in the solution. So what will happen if the hydrogen ion concentration would be high, it will go directly attack the carboxylic group. Okay, and it will convert it into COOH. Okay, that means the negative charge on the carboxyl group will be neutralized because it has accepted one H plus ion. Now, the same thing applies for NH2 group also. So, what will happen? It will be NH2 and it can accept H plus ions. Okay, because it's a base. So, it accepts an H plus ion and it becomes positively charged. So, that's why when pH is less than the pK value, the the molecule will accept the H plus ions and it will become positively charged. Okay, and when the pH is greater than the pK value, that means the H plus ions is low in the solution. So what will the molecule do? It will donate its H plus ion to the solution. So now what will happen in this particular case, if the COH is there, then that would release its H plus ion and will become a negatively charged moiety, and here it will donate its H plus and will become a neutral moiety with a lone pair. Okay, so this is how this concept works. Now, if we see here, in this case, in this particular case, the CO group has a pK of 4.25. This side chain has a pK of 2.19, and this NH3 plus has a pK of 9.67. So now we have to check at what pH all the three moieties would have a net positive charge. Okay, so in this particular case, a pH that would less than 4.25 would give a neutral charge on this COH. pH less than 2.19 would give a neutral charge on this particular side chain, and a pH that is less than 9.67 would give a positive charge on the amino group moiety. Okay, so when the pH would be less, then that would give a positive charge on this. So the pH less than all these three would give a positive charge on the glutamate moiety, the as a whole. So now the pH less than the, because this has the least pK value, so pH less than 2.19 would give a positive charge on these three, all the three moieties of the glutamic acid. That's why the pH, the options are wrong although, but the pH, if would it would be 2, then that would be the correct answer here. Okay, so now Is now the question clear? Please say whether it's clear or not. Okay, I'm considering you have understood the question, or else you can ask me at the end of this presentation. Okay, then moving on to the next question. Calculate the PI of histidine, and it also has three pK values. So, considering the concept that I have just told you, please calculate the PI of histidine and let me know the answer. Okay, any more answers? Okay, so yes, in this particular case also, what we'll do for PI, we'll select the nearest pH. So this would be 9.17 and 6. So the correct answer would be option number 4. Okay. So this is the only amino acid that have a PI that is near to the pH of our biological system, or the neutral pH. Okay. So it has various advantages, especially when it's present in the active site of many enzymes that are present in our body and functioning in the blood, or at nearly neutral pH. Okay. So this is the case with that. Now moving on to the next question. So we'll be starting now with the part of electrophoresis. So in the formula, force is equal to 6 pi eta Rn. Eta stands for viscosity, velocity, radius, or coefficient of friction. Can you please answer? Okay. Someone said first. Yes. So in this case, it's viscosity. Okay. So this is particularly the force that a particular molecule faces when it moves through a liquid. So when it moves through a liquid that have a viscosity, that is eta. Okay. So the force that is applied is equal to 6 pi eta r well. So r is the radius of the particular moiety here. Okay, so correct answer is viscosity. Now moving on to the next question. A DNA molecule having small size was ran on an agarose gel with a DNA molecule with large size. Okay. So the questions. Okay. So what would be the probable position of the large DNA molecule on an agarose gel? So would it be the near to the positive end of the gel, near to the negative end of the gel, in the middle of the gel, same as the small DNA molecule, or none of the above? Can you guess? Okay, someone said two. Okay, so for example, here is the agarose gel, this is the well, and here we loaded the DNA samples. Okay, so whenever we'll be moving the gel or the DNA bands would be moving on the gel, so what would happen? The DNA that is large in size would stay near to the well, and one would have the smallest size will move the farthest from the well. Okay, so this would be small molecule and this would be big DNA fragment. Okay, and whenever we load DNA onto a well, it would be near the negative electrode because DNA is a negatively charged protein and it will move towards the positive charge. So this is the cathode, sorry, this would be the anode and this would be the cathode. Okay, so the correct answer would be near to the negative end of the gel. Okay, and this would be the correct explanation of how the molecule travels on a agarose gel. So, as I explained you in the last question also, this is a molecule and it has a charge on it that is depicted by Q. So, when it moves in an electric field, it moves with the force of charge into the electric field. Okay, because we have also applied the external electric field here, that is E. So, whenever we are applying, so it would move with the force of Q, but it would also face a frictional force or the backward force due to the gel or the viscous medium that we are running it in. So, it would be equal to 6 pi eta RV. Okay, so here what will happen? Both these forces will equal out itself when the molecule will stop. So that would mean that 6 pi eta RV will be equal to Q and E. So here the velocity of the molecule, okay, so this would be constant. This would be also constant because it will be applied to the all the molecules present in a solution. Now eta would be the also constant because it's the medium. Only this would be variable. So now this is the charge on the moiety and this is the r or radius. This would depict the size of the moiety. So talking about the small DNA fragment, it would have small size and comparatively its charge would be more small. Okay, so keeping this ratio in mind, its velocity would be higher. Okay, and talking about the large DNA fragment, its size would be smaller. Okay, as compared to its charge. So its size would be bigger as compared to its charge. So that's why the velocity of a large DNA fragment will be less. That's why it will move slowly and will remain near to the well. and the small DNA molecule will move farthest and with a greater velocity. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So, an inert polymeric support is absent in which of the following types of electrophoresis? Moving boundary electrophoresis, vertical gel electrophoresis, 2D electrophoresis, or page? Can anybody answer? So, in this case, what happens? The correct answer is first, that is moving boundary electrophoresis. Okay, so this is the correct explanation. What happens in a moving boundary electrophoresis? We have a U-shaped tube in which the molecules are present in the form of a buffer. Okay, so this is the solution in which different charged molecules are present. Now, the electric field is applied in such a way that this is the cathode and this is the anode, and the different molecules having different charges will migrate towards their respective electrodes. But here, there is no polymeric substance present. It's not there. Okay, so that is why it is moving boundary electrophoresis, because here the boundary that we have. So initially, what will happen if we put on the liquid? Here, the liquid will be placed, which have different components, and this is the initial boundary of the electrophoresis. Now, as we apply the electric field, so what would happen? These molecules will move, and this boundary will also move. Okay, with them. So now the boundary will go up, and the next time the boundary will go up. That is why its name is moving boundary electrophoresis, because as the molecules will migrate, the boundary will go up and this would reach the respective electrodes. Okay, so this is the explanation. If anybody has any doubt, can ask. Okay, if not, then we'll move on to the next question. So now we'll start with the SGS page and how the polymerization takes place. So, what is the role of APS? That is ammonium persulfate in sodium SGS page electrophoresis. So, the options are: it is an initiator of acrylamide polymerization, it is used as a denaturing agent, it catalyzes the acrylamide polymerization, it is monomeric unit used for preparing gel. So, can anybody answer? Okay, someone said first. Any more answers? Okay. Any more answers? So yes, the correct answer is first. That is, it's an initiator of acrylamide polymerization. And how does it function? So this is the explanation. So what happens for a page to polymerize? There are two components required. That is the main component, acrylamide and bisacrylamide. Okay, so acrylamide and bisacrylamide to form a polymer. Okay, so to form multiple chains, it would require a free radical mechanism. And how this radical mechanism works? This is because the ammonium persulfate, that is APS. So APS provides the sulfate free radicals. Okay, so now what will happen? These sulfate free radicals interact with the acrylamide molecule and it forms uh, acrylamide sulfate free radicals like I have shown in this particular equation. So first, this is the sulfate free radical and it interacts with the acrylamide molecule and forms the acrylamide free radical formula. Then this particular free radical go and interact with other acrylamide molecules forming a chain. Okay, such like that it form multiple with multiple acrylamide molecules it form a chain, and then at the later stage it basically it also reacts with the bisacrylamide forming a cross link. Okay, so this bisacrylamide basically helps in cross-linking of the gel. So these, as you can see, these are cross-links between the gel. That is why a gel or a mesh-like structure is formed. Okay, so this is how APS helps in its polymerization, the acrylamide polymerization. Okay, so any doubts in the mechanism? Okay, if not, then we'll move on to the next question. So, what is the role of bisacrylamide in SGS page gel electrophoresis? So, it is a tracking die for monitoring the progress of electrophoresis. It acts as a cross-linker. It is a monomeric unit for preparing the gel, or it is used for providing negative charge on the protein. So, can anybody guess? Okay, just now I told the answer in the previous question itself. So, any more answers? So, yes, it's act as a cross-linker for the polymer. Okay, so just now I explained that bisacrylamide has this moiety. Okay, so this basically helps in polymerization or cross-linking between different acrylamide molecules so that it can form a mesh-like structure. Okay, so this is the role of bisacrylamide. Okay, now moving on to the next question. Pore size of a vertical gel electrophoresis can be changed by varying the concentration of acrylamide, APS, SGS, or TEMET. Can anybody answer? Okay. Any more answers? Okay. So yes, what would be the in case of page? We have two components that form the base of the gel. That is acrylamide and bisacrylamide. So, if we have to change the pore size of that particular gel, we have to vary this concentration. Okay, so the concentration of these particular components would vary the pore size. Okay, because these forms the base or the mesh kind of structure of the gel. So if they are in low quantity, the pore size would be more. If they are in high percentage, then the pore size would be less. So the pore size is controlled by acrylamide only. Okay, now I have a question for you. If APS is added more in quantity, then will it affect the pore size of the gel? Because it helps in polymerization of the gel, right? So if we add APS in the solution of this particular gel, would it affect the pore size? Okay, someone said no. Okay, can you explain why no? So yes, the correct answer is no only. It would. Okay, just okay. So what happens? It will not affect the pore size at all. Okay, but what will happen, because as we saw in the last image also, it only forms free radicals. Okay, so whenever there will be a free radical, the acrylamide would be active. Okay, 1 minute. So acrylamide would be active and it would cross link with the other acrylamide and bisacrylamide molecules. So it would not affect the pore size. Rather, if the pores, for example, if APS is more in quantity, okay, so it might affect the pore number rather than the size, okay, because it would allow more acrylamide molecules to cross link, and the number of pores that are formed in the gel might reduce. Okay, so there was one comment. So, sir, for polymerization to occur, we need cross-linking of bisacrylamide also. So, and acrylamide also needed. Yeah, for cross-linking, we need bisacrylamide, but APS is only needed for polymerization, nothing else. Okay, so varying the concentration of APS in the solution would not vary the pore size. Okay, pore size will only be varied only when the gel or the mesh-like structure that is formed because of acrylamide and bisacrylamide would vary. Okay, so this is the case. And APS, when added in more quantity or less quantity, that would affect the pore size of the particular gel. So am I clear, or I need to explain more? Was the explanation clear? So if not, then we'll move on to the next question. If you guys have any doubt, we'll discuss at the end of the presentation. Okay. So which of the following is used for providing negative charge to the protein for electrophoresis? The options are glycine, beta mercaptoethanol, bromophenol blue, or SGS. Can anybody answer? Yes. The answer is SGS, that is sodium dodecyl sulfate. Okay. So it is a negatively charged detergent. This is the structure of the SGS. Okay. So this is the detergent molecule that is basically have a structure of a fatty acid. Okay. So it has a long hydrophobic tail, and the head of the particular molecule is negatively charged because of the sulfate molecule. Okay. And it uh, attaches to the protein molecule and provides a negative charge on the protein. And two more information about SDS. Okay, it attaches with the C alpha, that is the anomeric carbon of the amino acid in a protein. And secondly, it attaches to two amino acid. That means one molecule of SDS attaches to two amino acids in a protein. Okay, so any doubts in this particular question? If not, then we'll move on to the next question. So Comasi Brilliant Blue, that is a dye, has same affinity for gel and the protein band. Okay, so it's the structure. There is no reason why it only binds two amino acids, but because of the structure of SGS, because it has a high hydrophobic tail and the amino acids that are arranged in the form, it the as you know the structure of the peptide bond, right? It is this in shape. Okay, so the size of SGS is such that it interacts with the two C alphas of the particular protein chain. Okay, so that's how it binds to two amino acids. That's the reason that has been studied till now. Okay, so moving on to the next question. It has more affinity toward protein band and less affinity towards gel, more affinity towards gel and less affinity towards protein band, or no affinity towards protein band and gel. So can anybody guess? Okay, so Comasi Brilliant Blue is a positively charged dye. Okay, and it as like in SGS page electrophoresis, what we do, we add a SGS molecule to the protein so that it will become a negatively charged molecule. Okay, so now this positively charged dye will directly attack or attach to this particular protein that has a negatively charge. Okay, so now this would attract the protein band. Okay, now can anybody answer the question? Sorry, my video call was cut. Yeah. So it would have more affinity towards the protein band and less affinity towards the gel. Okay. So answer is 2. And someone said about the non-specific binding. So yes, what we do generally is it would have some non-specific binding to the gel also. Okay, but that binding would be less effective and it can be removed easily. Okay, that's why what we do generally is that if we have to stain a protein band, we keep it in a staining solution. Okay, that would be containing high amount of Kumasi brilliant blue and the buffers that are needed for its interaction with the protein. Now, after staining for a particular time, for example, it might be 12 hours, overnight, or any other thing. It depends upon the lab and how and what we are staining. Okay, now we put it in a de-staining solution. So now this destaining solution will now put that particular non-specific binding. Okay, so it will remove this non-specific binding to a particular gel, and because this CPP dye has high affinity towards the protein band, will not be removed by this destaining solution, and it will be seen as a blue colored band. Okay, so is this clear? Okay, so now move on to the next question. Now we'll move on to the next question. So, what percentage of acrylamide should be used for resolving a protein with molecular weight of 5.5 kilodalton? It is 20%, 5%, 10%, or 8%. Can anybody guess? Actually, it's 20%. Okay, so the correct answer is lies in the molecular weight. So if we have to separate a protein with small molecular weight, okay, so this is considered as a small molecular weight. So now we would have a pore size that would be less. So we would use more amount of acrylamide. Okay, so remember this thing, when we have to use the acrylamide or agarose. the concentration or the amount would be inversely proportional to the pore size. If we increase the concentration of acrylamide, the pore size would be less. Okay, so this is a small molecular weight protein, and it would require large amount or more amount of acrylamide percentage. Okay, and if we need to separate a molecular weight protein that is in the range of 200, then we would require small pore size, so less amount of, oh sorry, we would require large pore size to move the protein through the gel, so less amount of acrylamide. Okay, is there any doubt in this particular question? If not, then we'll move on to the next question. So which buffer is commonly used in native horizontal page? Can anybody guess? So in native horizontal page, we generally use trisglycine buffer. And remember one thing, whenever we are preparing any gel, okay, so in any gel, we would use 1x buffer only, not 10x. Okay, so here in the native horizontal page, because it's a horizontal page, we would use 1x trisglycine buffer. And if it would be vertical page, then we would use tris borate EDTA buffer. Okay, so the correct answer is option number 1. And 10x generally we prepare as a stock. From this 10x we prepare 1x and we would use to prepare the gel. Okay, so moving on to the next question. The purpose of adding glycerol in SGS page loading die is to increase the density of the sample, to facilitate loading, to prevent degradation of the proteins, reduce the clumping of different proteins present in the sample, or provide uniform negative charge on the protein. So can anybody guess? Yes. The correct answer is option number 1. Okay, so glycerol is has high density. Okay, so what we require actually is, so consider this as a page. Okay, these are the wells that we form. So whenever we have to add a protein here, it would have high density, so that if we add from here, it would go directly down to the this particular region of the well. Fine. If we do not have that particular density, then it would, because here is the buffer. Okay, and we would require buffer for loading into the gel. And if we do not have had glycerol or any other substance that has high density, then the sample would flow out and move into the buffer rather than going into the into this particular well. Okay, so this is why we add glycerol, to increase the density of the sample. Okay, so moving on to the next question. The stacking gel has a pH of 6.8 for the proper stacking of polypeptides with different lengths. The statement describes most appropriately the mechanism of stacking is: uniform charge distribution of different polypeptides occurs at pH 6.8, which leads to stacking. Protein gel sandwiched between slow-moving uncharged tris HCl is in front, and fast-moving charged glycine residues. Or proteins get sandwiched between slow-moving uncharged glycine in front and fast-moving charged tris HCl. Or proteins run ahead of the advancing glycine tris front and get sandwiched while approaching the resolving phase. Can anybody guess? This is on the mechanism of how the stacking and resolving happens. So, any answers? Okay. So, the correct answer is option number three. Okay. So, how it happens, I'll explain. Considering this as a page, it has two components. This is the resolving gel, and this is the stacking gel. Okay. Sorry, sorry. I said the wrong thing. This is the stacking gel, and this is the resolving gel. Okay. So stacking means the protein is basically leveled up here so that it will be resolved in the next step. Okay. So they just want to stack it up at a level so that it will start resolving. And resolving means it will be separated here on the basis of their molecular weight. Okay. The pH of the stacking gel is 6.8, and here the pH is 8.8. Okay. So now what happens? The buffer that we use is tris, HCl, and glycine buffer. So it would provide the chloride ion, it would provide the glycine residue, and then we have one more component, that is protein, and it is also negatively charged. So, and for for information, the pK of glycine is 6.5. Okay. So what happens in case of the stacking gel? The protein is negatively charged because now it will not have any effect of pH on the protein because it is bound to SDS. Okay. So this negatively charged is due to SDS, and chloride is inherently negatively charged. Okay. So it will not have any effect of because of the pH. Now here glycine, it's because the pH is 6.8 and its pK or pi is 6.5, that means it will be neutral. So in case of stacking, what will happen? First chloride ion will move, then the protein, and then the glycine molecule. Okay, this is on the basis of the negative charge on particular molecules that are present in the gel or in the uh, buffer that we have. Okay, now there is a partition now which leads to a region that have a more pH. So the pH of the stack resolving gel is 8.8. Now what will happen? Chloride will have the highest negative charge. Okay, now glycine will move towards the from from neutral to negative charge. Okay, now what will happen? All the three moieties will have a negative charge, but now the movement will be on the basis of the molecular weight or the molecular mass. So now chloride ion will be moving first because it has the smallest molecular weight. Then comes the glycine residue, then comes our protein. Okay, so this is the mechanism of how the glycine, chloride, and protein will move in the stacking as well as in the resolving. Okay, so any doubts in this particular question? Sorry. If not, then we'll move on to the next question. Which of the following can be used for studying the protein-protein interactions? SGS page, urea page, native page, or denaturing agarose electrophoresis? Can anybody guess? Okay. Any more answers? So what happens exactly is, if we are using SGS page, so we are applying the protein with the SGS. Okay, the protein solution is with SGS. So SGS will provide negative charge on the protein. So all the proteins that are there will be negatively charged. So that means they will repel each other. So the protein-protein interaction would also be broken. Okay, so this cannot be used for studying the protein-protein interactions. Then we have the urea page. Okay, urea page will provide us with urea. So urea breaks the hydrogen bonds. Okay, so in some proteins, it's bound with the disulfide bridges, the protein-protein interactions that we can see. But in some cases, it is bound with the hydrogen bond also. Okay, so that would also break the bonds, or rather the interactions between two different subunits. So this will also be not used. Then we have denaturing agarose electrophoresis. So once we have denaturing electrophoresis, that means the interactions that the two subunits would have will also be broken. Okay, so this will also be not the case. Native page only allows us to study the protein-protein interactions, because here we will not be using any detergents or reducing agents or any other molecule that would affect the interactions of the protein. Okay, so we will not be using any three of these. So the correct answer is native page. Okay, so any doubts? Okay, so what happens in case of SGS page? We use SGS, that is a negative detergent. Okay, so if we are applying SGS to a solution of proteins, that would apply to all the proteins in the solution. That means the negative charge on the protein would repel the other protein also, because it is also negatively charged. Okay, that is why the protein protein interaction will not be able to take place in that particular gel. Okay, now if we have the urea page, so urea basically breaks the hydrogen bonds. Okay, so it will not allow us to study the interaction between different proteins. So if a protein molecule is interacting with other protein molecule through a hydrogen bond, okay, so in that case it would break those interactions and leave the proteins separated, so we will not be able to study the protein protein interactions. If we are using the denaturing agarose gel electrophoresis, so in that case we use certain conditions that would actually, in case of agarose electrophoresis, we do not generally study protein content. Okay, because it will not be able to resolve it properly. It is for the RNA gel. Okay, so we'll not use agarose gel electrophoresis for studying the protein amount or protein content. But in case of native page, we do not use any of such components, for example, SGS or beta mercaptoethanol, that is a reducing agent, or urea in gel itself, so that we'll be able to study that how a particular protein is interacting with another particular protein in a cell based scenario. Okay, So am I clear? Okay, moving on to the next question. What type of page is used for studying the fold states of a urea? Oh, sorry, fold states of a protein. It's native page, SGS page, gradient urea page, or agarose acrylamide gel page. Can anybody answer? So what happens in the real-time scenario when a protein folds? So this is the polymer chain or the polymer of amino acids, the protein. Now when it folds upon itself based upon different interactions, being it ionic interactions or van der Waals forces, hydrogen bond, and various other interactions that are non-covalent in nature. Okay, hydrophobic interactions. So all these interactions help the protein to fold upon itself and form a functional moiety. Okay, so now if we have to study the folding state of a protein, because there are certain states of a protein in which it's folded. So now if we apply a gradient urea page, okay, so urea basically breaks all these interactions. So it's a denaturing agent basically. So if we need to study the folding states of a protein, then we'll apply the gradient urea page. Okay, such that if we are using a low concentration of urea, that would break some amount of interactions, but some of the interactions would be still there. Then increasing the concentration of urea would lead to fully denaturing the protein, and then in between we can study different folded states of a protein. So here gradient urea page would be the answer for studying the fold states of a protein. So any doubts? Okay, if not, then we'll move on to the next question. So you are given a sample with complex mixture of proteins with negative and positively charged proteins. Which electrophoresis technique will you use to separate the proteins for further analysis? So it's a urea page, gradient urea page, native page, or horizontal page. Can anybody guess? The last question, right? So what happens whenever we talk about a protein? So this is the primary structure of the protein. Okay, so primary structure is basically a polymer of amino acid, and this drives the folding of the amino acids upon itself. That subsequently forms the secondary structure, and for the functional state of the protein, it forms the tertiary structure. That would basically form the active site in case of the enzymes. Okay, so now these foldings upon itself is due to various interactions that are non-covalent in nature. So this is ionic interaction, van der Waals interaction, or the hydrogen bonds that are generally formed in the protein molecule. Now What happens if we try to break these interactions and study the different folded states of a protein? Then we would better understand whether how this folding happens and what is the reason behind this particular folding or the fold that is formed in a protein. Okay, so we apply urea so that we can break these particular interactions and study the different folded states of a protein. And we apply urea in different amounts, that is a gradient, we form a gradient so that we can study the different folding states of a protein, that how it is being folded upon itself and how it the mechanism of folding takes place basically. So is it clear now? Okay, so this was the next question, and the correct answer is option 4, that is horizontal page. So why horizontal page? Because in case of horizontal page we only require the charge on the protein. Okay, so here is the positive and negative, and here you apply the different proteins that are there, and it will separate on the basis of the negative charge will have the positive proteins, and here the negatively charged proteins are get attracted. Okay, so to separate both the mixture of negative and positively charged proteins, we'll use a horizontal page. Okay, so any doubts in this particular question? If not, then we'll move on to the next question. So, which of the following is true for 2D gel electrophoresis? The samples are separated first on the basis of isoelectric point and then separated on the basis of molecular weight. Okay, so in case of urea page, okay, urea and gradient urea page, the proteins will denature. They will leave their actual state. So the proteins that are there will not be able to separate. Okay, and our motive here is to separate the proteins on the basis of their charge, not on the basis of how we are going to study it. Okay, in case of native phage, it separates the protein on the basis of molecular weight as well as charge. But here our motive is only charge, not the molecular weight. That's why we are using the horizontal phage, because in that case the molecular weight component will be side down. Okay, is this clear? Okay, so moving on to the next question. So in 2D electrophoresis, so in case of native phage, the proteins are basically separated on the basis of their molecular weight and the charge. Okay, but these proteins here in the question, the charge is different. Okay, but no, like the two molecules having the same charge can have the having different charges can have the same mass also. Okay, so if we have the native phage, then we would separate the proteins on the basis of molecular weight as well as the charge. But if two molecules that have these different charge but same molecular weight, we will not be able to separate those particular proteins using native phage. So that is why we are using horizontal page. Oh, is it clear? So in case of 2D gel electrophoresis, what will happen, there are two components. Okay, first is the charge component and second is the mass component. Okay, so the correct answer is first on the basis of isoelectric point and then separated on the basis of the molecular. Okay, so first the proteins are ran on a gel, that is also a page gel basically. So that is isoelectric focusing. Okay, so the proteins will be run on a gel that would contain ampholites. Okay, so ampholites basically forms pH gradient. Okay, as we have studied in the previous slides also. So at pi, the protein will stop moving in an electric field. Okay, so that's how, if we have to form a gradient of pH, that means from 0 to 40, let's say for example. So if a protein has a pi of 7, then it would end up having a band here. Okay, so now what we'll do, this was the first stage. Now we'll tilt the gel and put it in an horizontal direction. So this would be like this. So this is 90-degree angle of this. And then we'll run the gel in such a way that it would be separated on the basis of their molecular weight. Okay, so the second component is page, normal page. So now the proteins that have specific bands at their particular pi will move on the basis of their molecular weight, and then they will be separated on the basis of the molecular weight. So this is called as the 2D gel electrophoresis because it has two dimensions. First was the charge, that was on the basis of the isoelectric point, and second was the molecular weight. Okay, so is this explanation clear? Okay, let's move on to the next question. The complex protein mixture has a was separated by SGS page, and the RF values calculated for four protein bands present in the mixture are given below. So for protein 1 it's 0.33, protein 2 it's zero point 0.5, for 3 it's 0.16, and for 4 it's 0.83. So the protein band that is closer to the well would be, so RF basically stands for the refractive index. Okay, so it stands for, or how it's calculated is, the length the particular protein is moved by the total length dye has moved. Okay, so let's consider this as a gel, and you have loaded it with a loading dye. So that is bromophenol blue generally. So what will happen? We run the gel till the dye has reached this particular column. We are talking about page here. Okay, so the dye has reached this particular front. So this would be the length of the dye that it has reached. And now when we image the gel, we have reached, for example, this protein has reached here. So this is the length that the protein has run. So RF is calculated by the length the protein has run by the length the dye front has been moved. Okay, so based on this, what protein would be closer to the well? Can anybody guess? Okay, any more answers? So to answer these kind of questions, what will happen? If we can see a protein here for say, the RF value for this particular protein would be near to 1, because the different is here and the protein length that is moved is here. Okay, so it will be near to 1. So this particular protein, that is protein 4, has moved the farthest in the gel. Okay, so this is protein 4. Now talking about, then as we move from the farthest point of the gel to nearest to the well, the RF value would decrease. Okay, so here the RF value would be 0 and here the RF value would be 1. Okay, so now the protein 1 and protein 2. So protein 2 is zero point 0.5, that means half. So it would be in between both the well and the farthest one. Okay, then we have protein 1, that is 0.33. So it would be something around here. Okay, or maybe sorry, it would be something around here, that is just away from the middle point. And then we have protein 3, that is 0.16. That would be near to the well because it would have large size. Okay, and it would have moved very small length as compared to the different. Okay, so the correct answer is protein 3. So is this question clear? Okay, so let's move on to the next question. A protein has a molecular mass of 400 kilodalton when measured by a gel filtration chromatography. So for explaining gel filtration chromatography, it gives the actual molecular weight of the protein. Okay, so this 400 kilodalton is the actual molecular weight of the protein involving its subunits also. So when subjected to phase electrophoresis with SDS, so that SDS is also applied to the particular protein, it has the three bands, that is 180, 160 and 60 kilodalton. Now, when this is applied with SDS and the beta mercaptoethanol, that is a reducing agent, then three bands are also formed at 160, 90, and 60 kilodalton. What would be the subunit composition of the protein? So, the actual size of the protein is 400 kilodalton. When SDS was applied only, it forms three bands at 180, 160, and 60. And when SDS and beta mercaptoethanol are applied, then it forms three bands again, 160, 90, and 60. Okay, so what would be the subunit composition of the protein? Can anybody think logically? Okay, this can be a multiple selective question also. Okay, so beta mercaptoethanol basically helps in breaking the disulfide bonds. Okay, so what would happen considering this case SGS, first the SGS case? So here we can see three bands, 180, 160, and 60. Now, in the second case, when the disulfide bonds are also reduced, in that case we can see a additional band at 90 kilodalton also. So, if we can see 90 plus 90 is equal to 180. Okay, so now that means the protein has four subunits. Okay, so which means 160, 90, 90, and 60. So you can total it out, and this would form, oh sorry, so this would form 400 kilodalton. That is the actual molecular weight of the protein that was reduced from the gel filtration chromatography. Okay, So the protein has four subunits, and these two subunits that are there are linked with the disulfide bonds that has been broken by the beta me, that is beta mercaptoethanol. So option 1 as well as option 2 is correct. Okay, so let's move on to the next question. A mixture of glutamic acid and lysine is separated by paper electrophoresis in two experiments. So at pH 1 and at pH 12 respectively. Which of the following options is correct? The pI of glutamic acid is 3.22 and the lysine is 9.74. So this question is based upon the concept of pH and pL. Okay. So think logically and give the answer. We have discussed this part in this slide itself. So for your reference, when pH is less than pi, the protein is positively charged, and when pH is more than pi, then the protein is negatively charged. Okay, someone said 4. Any more options? Any more answers? Okay, so the correct answer is option 4. How? We'll explain. Now, considering the first case, that is glutamic acid, its pi is 3.22. Okay, so it has two carboxyl group and one amino group. Now at pH 1, first we'll see at pH 1. Okay, so pK value is around 4, around 2, and this is 9.74. Okay, so this, the charge on this would be 0 because H would be attached. Charge on this would be 0 because it would be attached, and charge on this would be plus 1. Okay, so the net charge would be plus 1. Fine. Then at pH 12, what would happen? This would have a negative charge minus 1, this would have a negative charge minus 1, and this would have no charge. Okay, because H plus would have been removed from here. So the charge at this particular point will be 0, and the final charge would be minus 2. Okay, now considering the case for lysine that has a pi of 9.74. So it has three groups. Okay, CO minus 1, then we have NH3 plus and NH3 plus because lysine is a positively charged amino acid or a basic amino acid. Okay, so now at pH 1, what would happen? This would be 0 because H plus would have bind here, then plus 1 plus 1. So it will be plus 2, plus 2. At pH 12, what would happen? This would be minus 1. Okay, this would be neutral, this would be neutral, so it will be minus 1. So now we'll see the options. At pH 12, glutamate will have more negative charge than the lysine. So yes, at pH 12, the glutamate would have minus 2 negative charge and the lysine would have minus 1 negative charge. So the first option is correct. then at pH 1 glutamate has less positive charge than lysine. Yes, so at this particular pH 1 glutamate has +1 charge, and at pH 1 lysine has +2 charge. Okay, so yes, the option 2 is also correct. So the correct option is option number 4, that is both 1 and 2. So anybody has any doubt in this particular question? These kind of questions are mostly and routinely asked in your competitive examinations. So if not, then we'll move on to the next question. In horizontal gel electrophoresis, the electrodes are usually made of gold, silver, platinum, or tungsten. So it's platinum basically. So what happens if we consider horizontal gel electrophoresis? This is agarose mostly, and sometimes we use the horizontal page. Okay, so what happens, because this is routinely used, the gold and silver are generally not used because they are too expensive to use. Okay, and we use generally platinum because of this oxidation, because of the oxidation potential. So whenever we are running any gel, so the platinum is the most suited element for these particular electrodes. Okay, so moving on to the next question. The staining mechanism of ETBR dye used in gel electrophoresis for DNA staining is that it binds to DNA backbone, it intercalates between stacked bonds, stacked between two DNA molecules, or bind covalently to the nucleotide base. So can anybody guess? So yes, the correct answer is it intercalates between two stacked bones, so that whenever we are for a particular example, this is DNA molecule and it is stacked between these particular bases. Okay, sometimes that is why it leads to mutations in the DNA also. Okay, and it changes the radius of the minor and the major group of the DNA, that has been reported. Okay, so this leads to the end of our lecture. So if you have any doubts in this week's discussion or this PPT, you can ask. So if no doubts, then thank you all for staying till here, and we'll close the session for today and we'll meet on the next Saturday. Okay. So thank you all. I didn't get the question. Okay. Thank you guys.